Nothing.
Yeah, it's recording now. Thank you.
Yeah. So I'll just share the screen. Let me know if you can see it. All right. Yeah. Okay. Yeah. So as Juliana said, I'm a neuroscientist. I got my DPhil in Oxford, and then I went on to do a postdoc in the University of Glasgow, where I'm still currently a research associate, although I've basically have one foot in academia and another one outside, let's say. I'm also currently a data scientist at a startup here in the UK. And I thought we could discuss a little bit on how we can make sense of data and how that is important. And actually, like, one thing that you should realize is that whatever you learn on methodologies and tools and skills that you acquire during the next few years on your studies, you can probably apply to different fields. Although you're focusing probably on bioinformatics, you will see that the tools are used basically in all different fields. And I think that's an important thing for you to know, that you can actually work in different areas, and because your interests might shift, or you might want some changes later on your path. And so I thought we could start a little bit on talking about data in research. So basically, I've been a researcher most of my life now, and I've done a lot of experimental work, and we can get a lot of data from experimental work, and we can also get from maybe databases where people have collected data already for us. So I think for bioinformatics, most of the time that's the case. Sometimes you do have to collect your own data, and that step is actually very crucial for you guys, or for anybody that's working with data. You will realize that once you start analyzing some data, if it's not very clear the way it's been collected, how it's been processed, and all of those details, it could affect your ability to analyze it, basically. And here I put on basically steps that are used in, let's say, data science nowadays, and you can also use on research. These steps are very similar in a sense. So you get your data, right? So you might get, I don't know, your sequences, or your genes, and also phenotypes from a certain database or something. In my case, I used to get like behavioral data, and I had to analyze it, right? So usually the normal process of analyzing involves basically just plotting maybe some graphs and analyzing, comparing means or medians, and the standard deviation of the different populations that you're working on. And that process in itself, you're already kind of like applying a model to analyze that, right? So when we say that we expect that our data is a normal distribution, you're already saying, like, I expect this model to be applied, right? So in a sense, you're saying that your data follows that model, in a sense. And then when you compare the means, you're basically comparing the same model applied to different populations, and then you can compare them. And there's also other ways of analyzing, which is basically using more complex models, and that is required especially when you have a lot of data and something that you cannot deal with. You have maybe like many different features that you have to make sense of. So you not only have, for example, in my case, I had like responses to a stimuli, right? So the the person was going to respond to stimuli. I had, for example, reaction times. And that is basically all of the data that I had. So that's very simple. I can analyze it and compare like means and things like that. But then when you have more features to analyze, you have a more complex problem, then that can get complicated. And this involves basically some processing of that data. You might need to clean it, prepare it, and then you can maybe find a model where you can fit that data through. And that model, what it's going to do, it's basically going to extract the most important information about that data. And then you can maybe get to some conclusions. Sometimes that doesn't happen straight away, and you have to go back and redo some of the process. Maybe you need more data, and so on. So we're gonna go through those steps a little bit. Please feel free to interrupt me and ask questions. It would be better actually if you do, because then we can kind of like have a conversation rather than just me being doing a monologue here. But so let's say we get some data and we want to analyze it. What we could start with is what we call as exploratory data analysis. So it's exactly what the name means. You're going to explore your data. And what kind of questions can we ask the data? So we would usually try to describe the data. So here, for example, we have a few features of this data set where you have a passenger ID, you have age, you have the fare they paid, and you have their sex, right? And here, this is like a summary table that you can get running a program like in Python or R, and then you can get a few of these descriptive statistics that are sometimes relevant, right? So you can get like the count, so the number of data points that you have, or mean, or the standard deviation. You can get the percentiles, and this is usually only for data that is quantitative, right? So if it's a categorical data, then you won't have those measurements. But then I can start to get to know my data a little bit better. For example, mean doesn't make sense for passenger ID, right? Because that's just probably a counter, right? But I can see that my passenger IDs go from 1 to 1,309, right? So that's the standard, the min value here and the max value. That has to do, of course, with the number of data points I have, which is also 1,309. Now for age, mean is more relevant now, right? So I can have a look at what is the average age of my passengers. They are around 30 years old, 29.5 years old, right? And how much they paid. So I can see that there is a maximum of 512. The maximum fare someone paid is that one. And I can see the minimum one, which is nothing. One thing that I might need to figure out is whether nothing, what does that mean? Does that mean that the person traveled for free? Is that because that's a passenger which is actually a worker, for example? So that's something that I could ask my data. So I'm trying to understand the data that I have, right?
Anna,
And yeah.
which data set is this?
So this one is what they call the Titanic data set. I don't know if someone has played around with this before.
Can you explain a little bit what it contains?
Yeah. So the Titanic data set is basically a data set that describes passengers on the Titanic, right? So you're gonna have names, you're gonna have surnames, like we said here, a passenger ID for that person, an age, a fare, sex. You're gonna have also like the cabin that they were assigned, which part of the boat was that cabin, and these details. And you're also going to have one feature which is called survive, whether the person survived or not the accident of the Titanic, right? So this is a very classic starter data set for machine learning, and people love to play with it and analyze the features. So that's why I took some of the figures are from that. But yeah, it's just to introduce this idea, right? So I have a complex data set that has, for example, like the cabin number, it has the cabin position in the boat. I have how much the person has paid, right? And all of that information, I also have whether the person survived or not, right? And so I'm trying to understand what kind of data I have. So for example, for sex, apparently you can either have a value of 0 and 1. I can tell this from just this summary, right? Probably 0 is female or male, and the other one is the other way around, right? So that's why you basically have a minimum of 0 and a maximum of 1, and you have some weird mean here, which probably shows us that there's more of one sex than the other one. Otherwise, the mean would be 0.5, right? I have a question from Jose.
Yes, I have a question. I have a problem that when you have to apply statistics in your data. I mean, as an example, there are some particular cases in which I think it's impossible. Like I working on structure protein predictions and something like that,
Yeah.
and I think I can't apply statistics on this case.
Yeah, so it's exactly, I mean, I'm here still talking about some simple cases, right? But still, like, I can here see some easy statistics like the mean and so on. But in your case and in many others, even in this case, I can't just look at the data and tell you like, oh, this kind of persons they are more likely to survive than other types of persons, right? Because it's a lot of data, and we humans have some limits regarding that. And so these statistics, like the mean and so on, they will help us up to some point, but they might not be enough, right? And so we will need some more complex models to deal with that. And we'll talk about it in a few more slides, let's say. But yeah, we'll go back to that question soon. So basically, I can also look at correlations between the different variables, right? So for example, here, I'm looking at a correlation between the weight and the height of people, right? In this case, I think it's not the Titanic data set, but just so you have an idea. You can see that there's kind of like a trend there, but it is noisy, right? So you could try and fit a line through that, and you'd have like an equation that tries to predict someone's weight based on their height, right? But that is not very straightforward, and you would probably have a huge error in there. But we could try to see if there's any of these variables that correlate well. So could there be a correlation between whether someone survived if they are male or female? Maybe there is, because maybe the tendency at that point in time was for men to try to save the females and the child and the children, right? And they would put themselves into more risk at that time. So maybe there could be some information like that we could explore. And this is just like the first step, right? I'm trying to understand the features that I have, how they look. So I could plot, for example, like in this plot on the right, lower right here. This is basically a plot between the number of females and males depending on their age. So I binned the value of age and into a few bins, and I can have a histogram of that. And from this histogram, I can tell that there are the age average for males is higher than the age average for females, right? And I can also tell that maybe the distribution of females is a little bit more skewed than the male ones. So the male one looks a little bit more like a normal distribution. And the female, I think they're probably more younger females than younger males, let's say, and so on. So this is what you're gonna start looking at the data, and you're gonna start understanding that. So for example, if you get a data set where you have certain sequences, the presence or not of certain sequences, and then you have phenotypes that are associated with that person, then how are they related? Can I find a relationship between a specific sequence, the presence of a specific sequence and a certain phenotype? You can start exploring that. And if you explore it by hand, let's say, it's going to take a bit more time. So you might need like the help of some more complex statistical models to deal with that. But before we get there, you might need to clean your data, right? So first we get data in a certain, for example, in a certain file. Can I open the file? That's something that you're gonna have to deal with. Is that readable? Can I understand what the file is saying? And do I have all the data there? Am I missing something? Am I missing just a few data points? Do I have all of the features for every sample, or am I missing some features for some samples? So if you're dealing with missing data, there are different ways of doing that. So, and you can also have outliers, right? So maybe you have on the Titanic data set someone that has an age of 200 years old. Can someone be 200 years old? That's very unlikely, right? So that is a data point that you cannot use. And I mean, if it's just one in very many, it might not matter, but I'm just using like a very extreme case, but you could have something that doesn't make sense, right? And so it's very important that your data that you're going to use has very good quality, or at least a decent quality for what you need, right? Because maybe I don't need to use the age if I can see that the age is very, let's say, dirty, right? And it's very noisy, and maybe I can keep that feature out and use the other ones. That's one option. There's another option, which is inputting some data. So sometimes you can substitute missing data with specific values. So you could use a median value, for example, to substitute some of the data. That won't change much your distribution, and that will keep it in like, you could still use it if it's not like a lot of the data that is missing. And here I also put this kind of like, how can I call this? It's like a data quality characteristics that you can look for in your data, right? So you need your data to be accurate, right? So like I said, for example, the 200, it's not an accurate data point, right? 200 years old, it's not something that it's really possible, right? You need your data to be as complete as possible. You need it to be consistent. So I can't have weird names for like too many different categories, or you have a few categories that are very specific, and some of the, instead of having a kind of like a consistent number of categories for a specific, for example, in the Titanic data set, if I have lower deck, medium deck, and top deck, and then I have some called deck 1, deck 2, deck 3, right? So you need to have it consistent. And you can, most of these things you can fix in a data set if it's not very good. You also need the data to be valid, right? So it needs to be valid in a sense that it represents reality, right? So I could have a skewed data set if I only have, for example, I was discussing this with Gepoliano, if we only have a data set with samples that came from white people, for example, can we actually use that data to create a model and apply that to a more mixed race samples, right? It would be a lot harder for that to actually have a good accuracy if we're training the model with data that is not necessarily representing the reality that we're trying to describe. And you also, your data also needs to have timeliness, right? It's that might not be the case for you guys, but for example, in my case, in my work, we have a model that runs every day and uses data that is collected daily. If that data doesn't come, then I can't use that data, right? If that data is empty, my model is going to give me a value that is not real, and that can make a difference, right? Yeah, Giuliano, you can.
I think you just put it in a different way than what I am used, but I think your way of stating it is very good. That is the representation of reality. So when we are biased in these, for example, in these and I think in for machine learning and for artificial intelligence, because these technologies are so common nowadays, it's a really important debate that we need to do. And if the technologies that we develop do not represent the reality appropriately, it's also the problem that we have with bias. And putting it this way, the representation of reality is a very good way to address this issue. I just wanted to ask if you could comment more about this, or if you're going to do so in the next few slides about ethics, because I think one of the purposes of this course is also to, well, I at least want to promote a debate and a discussion about bias and about diversity and representation. And apparently it has a very meaningful and important consequence for democracy, for representation in democracies and in the law and citizenship. I think that's very important, and putting it like a representation of reality is a very good way to put it.
Yeah. I'm gonna touch this on a few other slides, but I can add another detail on that. So for example, when at the beginning of machine learning and all of these models development, so for example, face recognition, right? People might have heard of that already, that when it started, they trained the models on a set of data. And then, for example, some people wouldn't get their faces recognized, and that's maybe because they are not the standard, what they call the standard kind of like gender or white person, and so then their face wouldn't be recognized by their phones, for example, because the algorithm was bad, right? It was badly trained, and so they had this issue. I also read one which is very, very absurd, and it happened with Google, for example, which is so they have, I think on their Google Photos app, they have a way of classifying the images that they see, and apparently for one person it classified Black people into a folder called gorillas, and that's really absurd. And you can see there that there's a huge error there, and that is something that they missed completely, especially because they didn't have this care of the training data set. So the data set that you're using to train your model is very relevant to the outcome of that model. But I'll also give us a few other examples, which is kind of like important for us to pay attention to that. later on.
Hannah just commented in the chat that she learned about something called ethics in the digital age. Thank you, Hannah, for the comments.
Yeah, so that is very relevant, and a lot of people are discussing about ethical AI, right? Because that's the great question of like we know that these can influence elections, right? And you guys in the US are gonna go through that now. We also have deep fake, which is all around us and can easily manipulate people because yeah, reality is not reality anymore, apparently. So all of those things are very relevant, and understanding how these problems can happen is very important for us that deal with data, right? So you don't want to think that you have a good result and actually it's not a good result, right? And especially in the medical area as well, right? Because if you're dealing with people's lives, you cannot tell someone, oh, you're very likely to have this disease, so you have to take this treatment, if that is not like, if you're using a model that not necessarily is actually predicting the disease that well, right? And yeah, so it's going to touch a lot of the parts of our discussion today. Well, once you have cleaned your data, you need to select some of the features that you're going to use for your models, right? So Jose said that he's using on something for predicting protein structure, right? So you probably have a lot of data from kind of like different information on, I don't know, connections or between the different atoms and amino acids and so on. So maybe that what kind of data you need to use is very important. So like which features you're gonna use. Can you use the presence of a specific sequence in that protein? Can you use how are you going to predict the next one? You're going to have some kind of information on what is the likelihood of someone having this sequence or that sequence. So what kind of features we need? So for example, for the Titanic data set, going back to our simple example, if I want to predict whether someone survived or not, maybe knowing where they were during the accident is going to be very relevant, right? Because if they were staying in a cabin that was far away from the crash site, the likelihood of them surviving is probably higher, right? So that could be like an interesting feature for me to use in a model if I'm trying to predict who is going to survive, right? Or if I'm trying to predict how much someone spend or like how the fare they spend is related to survival, then I need to use that fare feature in the modeling, right? And sometimes those are not so most models are only going to work with numerical data. And I think Cipollino also mentioned that you probably did some of that, like changing the type of data that you have. So you're going to have to change categorical values, for example, into numerical values. Or you might want to bin the values in a certain way. Because, for example, if I have a continuous variable, which is age, right? It goes from, I don't know, the youngest one, which is 5 years old, to someone which is I don't know, 80 years old, right? But maybe I want to separate them into more defined groups, right? So I could do like children, adults, and elderly people. In that case, I'm kind of like binning that data and categorizing it differently. So that these are all what we call feature engineering. So changes that we make to the features to adapt them to what we want the model to come like to process. And we could also add new features. So for example, I could have a model that is time dependent, and I might want to add the day of the week, right? So I could create a new feature on my data set that will give me the day of the week, so that I can see if there's any kind of like seasonality in the weeks, right? For example, so if the data is somehow changing, and it follows a kind of like a weekly pattern as well. Or I can even use some of the algorithms that I'm going to talk to you about to actually generate some of these new features. Because if I have a huge number of sequences, for example, I could try to basically join some of them together into groups and say, okay, this is a certain group, and I'm going to turn this into one single feature, and this other group of sequences I'm going to turn into one single feature. So for example, I don't know, for Jose, you can correct me if I'm wrong, because I'm not from the bioinformatics area, but I could, for example, see all of the sequences that of amino acids that will have some kind of binding site. I could classify them as one thing and try to create one feature that represents all of them in order for me to put that into my model, right? And so once you're happy with the features that you have, you can start to try and find a model to use. And usually people will try different models, and that's completely fine. You can try different models, because basically what you're going to have to do is you're going to have to choose one, and that is going to be probably based on a few metrics that we call. So how accurate that model is. And there are ways of you comparing different models, which is through these types of metrics. So for example, you could look for the size of the error for a specific error for different different averages or accuracies. So this is how you're able to compare the models and see if you could get a better model with different models. But then how do we actually get to these metrics, right? How do we find them? How do we know that our model is doing well or not, right? So this is a very important part. So when you get your data set and you're going to train a model on that data set, it's very important that you don't train your model on the full data set. So you're going to separate a little bit of that data set. You're going to hide it from your model, let's say, and you're only going to put through the model a few of your samples. So for example, if I had 100 samples, I could hide 20 of them, get the other 80, and train the model on the other 80. Once I've trained the model on those 80 samples, then I can try, I will get that model that it was trained. So when I say a model was trained on the data, it was basically that it has some weights that are going to be changed depending on the type of model that you have, and you're going to try to apply those to the data that the model hasn't seen, right? And that's what we call the training and the test data set. So the training data set is the data set that you give to the model to train the model. And then the test data set is the one that you kept hidden from the model. And now you're going to try and see how the model does on that data set. Does it predict well that data set? Does it separate the data set in a proper manner or not? So that is a very important question. So that's going to define how well your model is doing. So and this accuracy on the test data set or the error on the test data set is what you're going to compare between the different models. And there are different ways of doing this. I'm not going to go into very many details, but it's also very important to take very good care when separating those data sets, right? Because, for example, if I train my model on on, for example, part of my data set, but that part is only related to women. And then for some weird reason, I ended up with the test data set that is for men, for example. Will that model perform well on my test data set? Probably not, right? So there's a chance that it won't. It depends on what you're analyzing, of course, but there's a chance that it won't perform as well. What I'm saying here is basically that you want your data set to be balanced between the different features that you're using. So for example, here on this graph, this is the fair feature that I was talking about. And here you have a training set and a testing set, right? And you can see that the distribution of the fair for those two data sets is very similar. And that is something that you want, right? So you don't want your test data set, for example, to be concentrated only on the high fair samples, right? Because otherwise that might not get you the best results for your model. And it's not necessarily that your model is wrong, but it's basically that your sets are unbalanced, right? And then there's also another thing that you should take care when modeling, right? So if I get a model, so here, for example, in this cartoon, they are evaluating candidates for hire, right? Hiring a new employee, and they have some candidates for that spot. And they have an algorithm to basically try and classify those candidates. And this algorithm takes into consideration these factors, these features. So the educational background, the past experience, the recommendations, and then the interview performance, and the enthusiasm for developing and expanding the use of the deep AI hire algorithm. So these are the features, but then there is an error, as the person says there. An analysis of our new AI hiring algorithm has raised some concerns. Why is that? Because you can see that one of the features has a very large weight compared to the other ones, right? And this can be very problematic because can you imagine, like, if you're in a company and you're trying to hire someone new for a specific job, but your hiring algorithm is somehow keeping out all of the Black females. You need to understand what your model is doing. You need to understand which features your model is relying more, right? So which ones are the features that are more important for your model, because that can tell you whether that model might be biased towards something that is not relevant. So again, something that will not be in accordance with reality. So far, okay? Any questions? All right, so now we're going into the machine learning part. I mean, all of the things that I'm discussing now already has something to do with machine learning, right? Because what is machine learning? Can anybody offer me some kind of description of that, or any hint of what you think machine learning is? Don't be afraid to be mistaken.
It's like using statistics to train an AI, maybe.
One of the things, yeah, you can use some machine learning algorithms to train an AI, yeah, for sure. So yeah, it's basically using some statistical models to train to basically generalize unseen data from data that was used for training, right? So these are models that are able to learn from a big set of data. And then when they encounter new data, they should be able to perform well on that data as well, right? And that is the great thing about machine learning is basically that the computer can learn from the data some importance, can process the important part of that data. And then when it sees new data on that same topic, let's say, it can recognize that, right? And it can point us in that direction. And there are some different algorithms that you can use, some different statistical models. And they are basically classified into three areas, which are supervised learning, unsupervised learning, and reinforcement learning, right? And the difference is mainly to do with how you deal with that data. So supervised learning, the algorithm is going to learn about that data, and that data is labeled, right? So I could give, for example, the algorithm a few pictures of cats and dogs, and I can tell the algorithm, this is a cat, this is a dog, this is a dog, this is a cat, this is a dog. So I'm labeling the data, right? So each of those images has a label. And then the algorithm, whatever the algorithm you use, is going to try to capture information from that image and associate with that label. So it's going to capture a few important bits of that. So it could capture that the cat has a bit more round face or something like this, or has the whiskers that way, and the dog has the canines more profound, like pronounced, and all of those things. It's going to capture that information. And then when I present new data, so I give it one photo of a dog that it hasn't seen yet, it will be able to recognize that. Now, with unsupervised learning, you're not labeling the data. So I could give the same set of pictures of the cats and dogs to the algorithm, and I could ask it, can you separate that into two groups? And then you would try to separate them into two groups, depending on those features as well. But for example, if I give only pictures of dogs and cats that are black and white, it might separate them into the ones with more black and the ones with more white, right? So it is a little bit less controlled in that sense, but it will try to do that separation for us. So that's an unsupervised learning, right? So it will just try to extract the important features from that. And it could be that they are not exactly what we're expecting, but that could be also important. We'll discuss a little bit later. And then there's a third one, which is reinforcement learning. And reinforcement learning, it has to do with reward-based training, right? So you can think of in a way that it's a trial and error, like we learn, right? So the algorithm can learn the way we learn. So if I eat something that is bad for me, then that's going to make me sick. And so I'm going to learn that makes me sick. But maybe I don't realize it yet, and I eat it again. Then it makes me sick again. And so there is some kind of like either a reward or a punishment associated with a certain action. And that is used to train a lot of AIs, for example, the ones that have to do some kind of decision making. And that is another one. We're not going to go a lot into the reinforcement learning part, mainly because yeah, I don't think it's that much used in the bioinformatics world, but you can have a look, you can be curious and just search for it. Maybe you'll find a paper that actually has used that. It could possibly also be used, for example, in Jose's problem, because you're trying to basically find some kind of connections between proteins, and you could train an AI to basically try and figure out the best approach for that. But yeah, so we're going to supervised learning algorithms. So just a very, very rough introduction to that. So you can have mostly those two types, regression algorithms or classification ones. And the regression algorithms are going to try and basically fit some kind of curve to that to differentiate or to be able to predict a certain value depending on another variable, right? So for example, here on this plot, I can fit a line here where I'm relating the number of TVs with the number of with the amount of sales that a store has had, right? And with this regression here, I can tell like how much that store is going to have in revenue, depending on how many TVs they sell, right? That is a very simple one, and the linear regression one it's usually like the most simple one, and is also a good one for you to always try, because it can be kind of like a baseline model depending on what you're dealing with. And you can have an error associated with this model, for example, that has to do with this distance from each point to that line that you drew there, right? So there's going to be an error associated, and how well that line explains all the dots. And then you can have logistic regression, where you can have, and here, of course, it's just a two-dimensional linear regression, but you can have it multi-dimensional as well, right? So it's just a linear combination of the different features, and you can have it in multiple dimensions. Then you, rather than imagine it on a 2D plane, you have to imagine in like a kind of like a 3D space or 4D, and then we can't really imagine anymore. But yeah. And now, for example, logistic regression, it's going to try and get some of the features. And in this case, for example, we can get the user age, the user income, and the user gender, and indicate to you whether someone with those certain characteristics are likely to make a purchase or not, right? So this is very used, for example, in either recommendation engines or in like stores like Amazon and so on. They can use models like this that are going to basically indicate whether someone is likely or not to make a purchase, depending on the information they have about that person. And then these are usually called supervised learning algorithms, because you have a specific value associated with a specific label, right? So here, for each value of the TV, you have a specific number of sale amount that is associated with that. And then with the logistic regression is the same for each purchase. For example, like if someone clicked on a link and actually bought the thing, they will capture the information about that person, that user, and they will save it. Okay, this person clicked on that link and bought it, or this person clicked on that link and didn't buy it, right? So this is the label that is associated with the other features that you have, the information that you have. And now for the unsupervised learning algorithms, you're going to have either what we call clustering, which is a kind of like a classification, or what we call the dimensionality reduction. And the clustering you can think of just by grouping, right? So what you're trying to do is basically, depending on the different features, you want to try and bring together all of the samples that are most similar between each other, right? And this will kind of like create a certain number of groups. And here, of course, like in this one, you have, for example, one feature here on one axis and another feature here on the other axis. And then in this case, we can clearly see there are 3 groups, right? But sometimes it's not that clear, right? And this is where clustering algorithms are going to help. They are going to help us identify these groups. And this is one of the kind of methods that I mentioned before that you can use in order to change your features, right? Because if I can group some of these features together, like in this sense, then I can reduce the number of features I have and make it simpler for my model. And then you also have dimensionality reduction, which is basically what it says. It's just trying to reduce the number of features of your space, right? So if I have, for example, age and height and weight, and then it will try to get all of those features and find some kind of like similarity between them where they are most correlated, and it will keep only that direction. We will transform those data into a new kind of like component, and you keep that component in order to describe that data. Yeah, Gepolino?
I was just going to comment that dimensionality reduction is a big problem in genomics because the students are going to remember that the one data set that we are analyzing, the neuroblastoma data set, has like 25,000 genes that we can analyze the expression of. And it's a big problem to decide between those. For example, it's a big problem to decide among those 25,000 genes that we are dealing with, which ones are the genes that contribute, for instance, for the in neuroblastoma, the high risk label that we looked at before, guys. So this is one technique. This is how I came across machine learning, I think, in the past, because this is a technique to help us decide, or at least we have a method that can help us decide which of the genes or the groups of genes are going to have a bigger impact, for example, in the risk category in neuroblastoma.
Yeah, exactly. And I mean, we also faced a similar problem in neuroscience as well, because we can, for example, we can image and you can get the basically the neural activity of several neurons, right? So hundreds of neurons as well, and thousands depending on the technique that you use. And basically the idea was to use dimensionality reduction to group together the neurons that have a similar response, so that we could more easily correlate that with behavior, for example, right? So it's a kind of like a similar problem, and that's why this one is sometimes used before you use some of the other kind of like algorithms, so you could do a dimensionality reduction on your data set of genes, right? That will somehow tell you these groups of genes are kind of like related, and we can group them into this component. And then you use that component for your modeling, and you use that as a descriptor of your data, so that it captures most of the important information in your data. And that is what you're going to use to train your model, rather than the whole set of genes. And that is very relevant indeed. And yeah, it's these ones are especially good for that. They can also be good as a kind of like a classifier, especially the clustering. You can try and use clustering for classifying different samples into certain groups. The only question then is, for example, once you group them, can you actually make sense of that group, right? Can you understand why they were grouped together, and see why that is relevant? So those are things that you have to keep asking yourself during all of this process of getting data, modeling it, and then extracting information, insights from it.
Anna, we have now 1 hour of discussion. We can do a break or a bio break now if you want, or if you prefer, we can continue. How do you want to do it?
It's up to you guys, really. Yeah, we can probably, maybe I can just do one more slide and then we can do a break, because I think that will be a nice break then.
Sure.
Yeah, because the last thing I'm gonna talk you about is just tell you about is neural networks. So these are part of machine learning, and you can find neural networks being used as supervised models or unsupervised models. So you can have both. And you can also have them in reinforcement learning as well. But they are basically they are based on our neurons in our brains, right? So as you might know, we have millions of them in our brain, and they are connected through axons and dendrites, and these connections have different weights, right? In a sense, because there are stronger connections and weaker connections. And in our brains, they are quite plastic, right? So they change a lot. But in neural networks, they basically change their weights. So the way we imagine neural networks is you have different nodes, right? And they are somehow connected. And you can have, for example, a very simple one, which is the one on the left here, which basically has kind of like a one neuron network. And this neurons, basically what it does is receives some inputs, and these could be our features that we've been talking about, right? And then they weight these inputs and sum into some kind of like a function that sums them together using those weights. And then depending on the threshold, it will give you one response or another, right? So this is a very simple network that you can have. It's like they call it the perceptron network, and it can be used in many different applications, and it can be used as kind of like a baseline model as well, similar to linear regression when you're talking about neural networks. But you can also have more complex ones, like the deep neural networks that we hear a lot about, or the RNNs, which are the recursive neural networks, because you usually have an input layer, you're going to have some hidden layers, and then you're going to have an output layer, right? And this output layer will depend, for example, on the type of output you want. It might just be one neuron, it might be several neurons or nodes, depends how you call them. But yeah, and they are interconnected, and they can have recursiveness so that they can influence themselves, or they can also have some kind of back propagation so that the information goes back into the network. So for example, if you're training, you can have models that self-train themselves, right? Because they are going to inform the previous layers whether that response, the end response was a good one or not. So this is basically part of machine learning as well, and it can be used in a similar way as the other models that we presented before. Yeah, and I think we can maybe do a break now, and we can talk a little bit more on some of the applications. So I can tell you a bit more on how I've used machine learning, and maybe we can discuss a bit on how we can use machine learning in bioinformatics as well.
All right, thank you. Let's do a 5 minutes break or bio break, guys. We'll come back in 5 minutes. Anna, thank you so much. This is amazing. I don't know if anyone that's coming back may have any comments or any questions. Yes, you can continue on. Thank you.
Sorry, I was just reading some of the comments. Yeah. Yeah, so Jose was saying that AlphaFold uses a neural network. Yeah, so that's exactly. Yeah. So regarding Kanav's question, so there are different ways that they are going to group the data together. Some of them, for example, they define a measure of distance, right? So that can be defined as a vector between two of the dots on your feature space, right? And you can calculate that distance between the different dots, and you're trying to basically group together the ones that are closer together regarding with that distance measurement. It can be easier to imagine in a 2D space, but it works kind of like the same way for multidimensional spaces as well. So it has mostly to do with how you define that measure of distance, and you can do it in different ways. And of course, different algorithms are going to do it in a different way. So for example, if you use decision trees, they are basically going to start grouping together samples depending on answers that you're going to have to specific features, right? So you could ask the question, is this gene more important? And then is this gene present? And so you put one group to this side and one group to the other side. And then you look at the second gene. Is this gene also present or not? And so that's going to separate one of your groups into two more groups, right? So it depends on the algorithm a little bit. I think Gepoliano also can point to what one of the articles we discussed. I think they use hierarchical clusters to basically separate some kind of like some of the kind of expression of genes or something like that.
neuroblastoma data set?
Yeah, I think that's the one.
Oh
Yeah,
yeah,
so
we commented on that yesterday, and that's something that I was planning to. I can find the link and put here for people. But in neuroblastoma, guys, I told you that there are two main cell populations, right? The adrenergic cell population that looks like neurons, and the mesenchymal cell population that is the population of cells that are undifferentiated. And so these mesenchymal cells are really dangerous because they do not die in chemotherapy. Chemotherapy cannot kill them. And I didn't tell you this part yet, but what my research is suggesting is that hypoxia has an impact in making the adrenergic cells become mesenchymal-like. And so all of that to say that hierarchical clustering and PCA also has been used to separate these populations. And so it is this machine learning method that was used to differentiate the two cell lines. And so what this has allowed us to do is to basically detect or diagnose or tell what expression patterns are the expression patterns of a population of cells that is really, really aggressive and can be leading to cancer relapse, because these mesenchymal cells, they can become differentiated and start growing again. So machine learning has allowed us to tell what expression pattern is characteristic of a adrenergic cell that looks neuronal-like, and the mesenchymal cells that are undifferentiated cells. So that's one of the reasons why I'm interested in these methods because they can allow us differentiate these populations of cells that behave very, very differently.
Yeah, and I think I actually added that into one of the slides as well, that paper. So regarding using how we use machine learning, right? I mean, here are some of the examples I've faced rather than how we use, but just to give you an idea of different ways that you can encounter uses for it. So for example, I've used on my research for identifying specific features on images. So you can have an image of a neuronal recording, right? And then you're gonna try and identify their neurons, and you have a fluorescent marker that basically changes, the light changes with time, right? And then I've used some machine learning algorithms to basically extract that and turn that into kind of like neuronal activity. So like spikes, kind of this different, it's a method basically to extract that information from an image, right? So turn the image information into numbers, basically. Or one of the companies I work for, they use images from the streets. So like they put a camera on the cars and they monitor the pavement, right? So they image the pavement, and they use neural networks to identify holes on the pavement so that they can properly resource maintenance to specific areas of a city, right? So you can use that in order to use better the resources of the town hall, for example, to work on the streets that are needing more of that kind of like pavement maintenance, right? And covering the holes and everything on the streets. And you can use it with different for different things as well. So you can also use those cameras to identify areas that need grass cutting, or so you can use these neural networks to identify certain features on the images that are relevant to what you want to do, right? And then I had also, so I've told you about how we were identifying groups of neurons that had similar activity and how that could correlate with behavior. And we can also, for example, just by looking at the neural activity, getting all of those informations, try to identify whether at that point someone is looking at a certain cue or another cue, right? So you can try to classify the activity, whether it's related to someone which is experiencing one sensory cue or another sensory cue. So all of these kind of things. And then also, and this applies to several areas, you can try to predict a certain indicator from the available data. So you could do that for a company performance. So you have some data on some company, and you have a specific performance indicator that you want to know how that's going to work, like predict how it's going to be on the next month or on the next year or so on. And you can use past data to basically try to predict that. And a recent project that I've been working on is basically using data from different factories of cement to predict so that it uses the data from the production of the cement to try and predict how well this cement is going to be resistant to a certain compression. So different cements have different resistances to compression, and they, in a certain construction, you can only use one type of cement. And the problem is the only way they know that is if they do a test basically 25 days after the cement is produced, right? And so they have to wait that long in order to sell the cement. And there are some other informations that you can get. So this is a project that is working with the cement factories and using these different machine learning models to try and improve the production process so that you don't spend a lot of resources. Also, you don't produce a lot of CO2, you don't produce excess residuals, so that you have a more efficient process and you have less of an impact in the environment as well. So we're using machine learning also in the cement compression resistance prediction, but also trying to predict the emissions of the different gases that are involved in our environmental crisis. So this is one. So these are some of the. It's not an exhaustive version of it. I can also tell you that we've also used to basically track behavior. There's a very cool development. It's called Deep Lab Cut, and you can probably have a look at that as well. It can basically track movement just by training a neural net in using a few data points, right? So you can teach a neural net to basically track movement. You can track even fine movement, like something like just playing a piano or something like that, with and then you can transform that into basically data that is coordinates and so on. So you can I could probably put it up here if we want later on an example with that. But yeah, I think you've also encountered a lot of the different uses of machine learning and AI in your life. So that's not very exhaustive, but regarding bioinformatics, I found a couple of papers other than the one that Gepoliano already mentioned, which is, I think, this one, the super enhancer differentiation states that used machine learning in order to get to their results. So you can predict the type of cancer, for example, using just RNA sequencing in using a classification algorithm that is basically machine learning as well. And there's some neural networks as well that can deal with metagenomics, and as even Jose alluded to, there's a lot of softwares that are using these to basically help us with our research and help us understand the data that we have. I don't know if anybody else remembers another information on that. I'll just check the message. Yeah, okay. Anybody has any more examples on bioinformatics? It's not really my area of expertise, but you probably have read some papers, or you're going to read a lot of them that are going to use some of these more complex statistical models and to try and solve different questions in bioinformatics. But yeah, I think that was basically it from me at least. And I'll just leave with this one. As I've been mentioning, like, we have to be very careful with data. We have to be responsible and ethical with the way we deal with data from the beginnings, from the collection point where you're going to collect data that is sensitive. You need to treat it accordingly. You need to hide the identification of people, because that might be personal data or something like this, to the point of what kind of data you're going to use to train your models and so on. So yeah, I put this cartoon again. It's just, I think kind of summarizes that idea that if it's good that we realize, okay, we don't have good enough data and act on it, but not the way it is depicted, shown in here. So yeah, it's just be careful with the way you're going to deal with your data. That's the idea. It can have big impact in the world.
Anna, thank you so much. This was great. Thank you very
Yeah,
much.
thank you for changing the time. I didn't
Thank you
thank
the
that,
students that accepted to do that.
yeah, exactly.
Time is it
And
now for you?
if anyone has questions, if you don't want to ask them now, you can ask them later. Gepoliano has my contact details, and I'm happy, yeah, if you need to talk about machine learning, or if you want to talk about, yeah, life, it's fine. We can always try to help.
Thank you. That's great. As you were presenting and as we talked before, I didn't do the exploratory data analysis on the neuroblastoma data set that we are analyzing. That's certainly something that I'm going to implement for the next versions of the course. And everyone, so that you all know, either Friday or Monday, we are going to do a machine learning practice to classify neuroblastoma in the high risk or non-high risk categories. That's why I thought that Anna would be a great fit to get us started in the machine learning world. I just thought of maybe a last question. Could you use another type of prediction algorithm other than the neural networks, or are they the best? Are they the best that fit the needs in the samples that you have and the work that you do?
So they can be, like, they are usually more accurate than other ones, but the problem with neural networks is that sometimes you can't really tell what they are doing. So it's kind of like the question that kind of was asking, like, do I know how they separated them into those categories? It can be tricky to understand how they did that. Like, it's harder for you to know if that algorithm is weighting more one feature than the other. So and sometimes it can be a little bit overkill, right? So if you have very well correlation, like a linear correlation, probably a linear regression will be enough, right? But if you have more complex data and a lot more data, then usually neural nets are better at dealing with the information. I think they are also quite better at dealing with visual information as well.
Okay. Thank you very much.
Thank
All
you for the presentation.
Thank you all for listening.
Does anyone else have any questions for Anna? Thank you. Thank you very much, Anna. All right. I think right now we can switch gears a little bit to talk about your guys' presentations on Friday. We only have 30 minutes left. I put some information in the course handbook. You were going to so you can see there that I included some notes about your presentation on the assigned R operator or the command. I should not say a program, not at this point. Let me share my screen. Okay, does everybody know how to access the handbook? You go here to the the shared drive, the bioinformatics the BBC, the bioinformatics bridge course 2024. We have here the bioinformatics handbook. You just open that. And there you go. And so this first point here, I hope you guys, okay, I can show you that. There's a list here where I have assigned each one of you, if this opens up. The list, this is the list. I assigned a command to each one of you. So the idea here would be that you talked about that command for 5 minutes. And I put here some slides. Right? This is an example of slides for presenting the command head, for example. It's a command that we use it. So one slide showing, one slide defining the command. You can use technical language, but also non-technical language, explaining what the command does, right? So for example, here, I put the head command lets you see the top lines of a file. And then it prints 6 lines of the data frame. To specify a number of lines, we need to use the n equals 10, or n equals the number that I want of lines, right? And so in the second slide, it's going to be one slide with an example of how the command is supposed to work. So here we did this example. You can do something similar. And then one slide giving examples of mistakes that can occur with a screenshot of the error. So that's what is in the next slide. Okay, do you guys have any questions about this or any comments? So by Friday, you should be giving a 5-minute talk about this. Okay, now if you can choose something different, you can choose to explain a figure, right? And if you choose to explain a figure, I'm gonna close this. If you decide to present a figure, it could be a figure of a paper, a science paper. So I put here this example of this paper because probably when we do the terminal next week, we're going to do a variant call using these mitochondrial sequences. So you show the title of the paper that you chose. And by the way, I expect it to be one of the papers that we have here. I would accept you guys to share about your own paper, but for example, for next week, we're gonna have a presentation again next week and the week after. I would like it to be on one of the papers that are here, because these papers are about the material that we cover in the code that we do. So this paper is the ancestry of Brazilian mitochondrial DNA lineages. So I show the title of the paper, and in here I describe why the paper or the figure is important. So we would give a slide explaining why the paper or the figure is important. And then the figure itself, what does the figure show? This is in this case, this is a phylogenetic analysis of mitochondrial sequences. And by the way, this information is also in the handbook. One slide showing the title of the paper, one slide to describe why the paper or the figure is important, and then the next slide should be a slide to describe what the figure shows or how that knowledge impacts your life or science in general. So in this slide here, why is it important basically? Or why does it impact your life or science in any manner? And I think because we are going to discuss a little bit, I'm going to state why this particular figure is important. In my case, I am Brazilian, and one of my goals is to integrate genomic information into the Brazilian health system, which is public. And by the way, I should mention that there is a genomics project going on right now in Brazil that aims to sequence 100,000 Brazilians. And so that's why I speak about this figure, because it is important to illustrate that concept. And then the last point is one slide for the conclusions, if there is any conclusion. And so my conclusion here is that I have access to genomic sequences that were originated in Brazil, and I can use to integrate to publicly available data because this data is already publicly available. And so I can integrate that into something that is already publicly available. I wanted to know how you guys feel about this, because for me it is not really challenging, but I know that, and it's okay if it is, if you perceive this as something that is challenging to do, but I would like to hear from you if you feel like this is something that is challenging, that is too much for you to do, because yeah, I just want to hear from you guys. Can people share their thoughts with me about this? Alexandra, what do you think about this proposal?
I think it's fine. It's good for me.
Advaita,
Oh yeah, I'm good with it.
you're good?
Yeah.
Victor?
Yeah, I'm fine with it too.
Okay, do you prefer the figure or do you prefer the commands?
I probably pick the command.
Okay.
Yeah.
Eric, do you hear me? Was the
Yeah, sorry, I was just having some trouble with the mouse.
okay?
Yeah, I think it should be fine. I think I'll end up choosing the command because I think it's just a little more straightforward. But I think the presenting a figure wouldn't be challenging unless someone doesn't have like too much practice reading papers and all that.
Okay. Does anyone else want to talk about this? I just want to feel how you guys feel about this, because I do not want to give you something that you think it's not okay to do.
So I had a question. On Friday, after we present, we're also learning about machine learning as well, right?
Yes, that's the plan. Yes, that's the plan.
And then when we're going over like those lessons about machine learning, would we be using these same data sets or like these same studies?
We are going to be using the neuroblastoma data set.
Oh, okay. All right, sounds good.
Mm-hmm. All right. Any other questions? No questions? Okay. The last part of this is explaining a figure that we have plotted, and that's what I call here the project. That's what I call, I should put in parentheses here, the project, or the mini project is how I called it. I am putting it under the, gonna close this, under the umbrella of a project, but here, really what I'm talking about is the hypoxia project, and the figures that I put here does not convey that. But it's okay, I can change that later. So let's see here. So one slide, I would like you to do the syntax for the plot that you generated. One of the figures that we worked together, or that you got to plot by yourself independently, but from the data set, from the, so far from the neuroblastoma data set, right? So here, for example, one slide for the syntax. So I'm presenting the syntax of this qplot function. We already discussed this a few days ago. And then one slide to summarize the message of the figure. So you show the figure. So here, the summary here, one way that I can present this is I can say that the high risk group is composed mainly of patients with high MECAN expression, or that MECAN expression, or that this is a MECAN expression in the high risk group. And one summary, or maybe it's already a conclusion, is that majority of the high risk patients have MECAN amplification. And so, so one slide to describe the figure, one slide to describe the figure, and one slide for the conclusions. So if there are any conclusions, you put it in this slide. So in this case that I just discussed, MECAN expression accounts for a lot of high risk characteristics. Why? Because we see here that the high risk group has a very high MECAN expression, whereas the non-high risk group does not have a lot of MECAN expression. And that would be it for presenting on Friday. Questions or comments about this? All right. So the other thing that I'm gonna show now is I'm going to describe a scientific paper. Has anyone of you read a scientific paper before? Okay, Alexandra said that she has read. I mean, probably read is not a very good way to ask this question. Or, okay, Eric also has a few times. Are any of you guys involved with research? Okay, a good number of you have. Okay, so maybe I should not be so concerned about guiding you. I don't know. I am also asking you guys this question. If you guys think you would benefit of me guiding you through strategies to understand a scientific paper, maybe. I would like to hear that from you, if you can answer that. Okay. Yeah, okay. Alexander said yes. Yeah, okay. Okay, I didn't have this in mind. How can I say this also? I did not work on that yet. Okay, Kanav said I'm not super familiar with reading through scientific papers. Yeah, and I do not expect, for example, I myself, the first time when I had to deal with a scientific paper, I was in the 8th semester of my major. And so that would be almost 4 years of an undergraduate major in Brazil. So I did not get in contact with scientific papers as a freshman. But so the reason why I'm doing this today, and we can take the next 15 minutes or so to walk you guys through this paper, is because we need to talk about the subject. We need to talk about the paper, and I think that this paper that I'm going to talk about now really conveys important concepts. So let's take a look here. Let's take a look here. So this paper is in the papers folder. It's the BAG 2023 paper. So this paper is this is basically a discussion on a paper about the genomics analysis of Beethoven's hair. So what I try to do here is I try to show you guys important questions about the paper. And the reason why I think these questions are important is because of my personal interest in this subject area, and also what I can so far extract from the information that the authors are providing me with. Okay, and that's the template that I use it to generate the information that I'm sharing with you right now. We do not need to understand all the details of this paper right now. I'm just giving you a very high level, and probably I'm going to modify these slides in the next couple of days. So in the first slide here, I'm showing a picture of the paper, and it's always a good practice to read the title of the paper, because the title gives you an idea of what the paper is about. Basically, it summarizes the idea of the paper. And this one paper is the genomic analysis of hair from Ludwig van Beethoven. It's interesting. So this is the BAG 2024. I do not see the year here, but this is the author. One thing that is interesting is that this is the first author is from the Department of Archaeology at the University of Cambridge in the United Kingdom. I like this is a Cell paper. I like the Cell papers because they have graphical abstracts, and I think this is really useful. So they highlight here 4 points, I would say, about the paper. The first one is the genetic ancestry of Beethoven. Right here, they are telling us that he has more than 99% of European ancestry. That information is good to be there, because you're going to see in the next couple of slides that there is debate about his ethnical and racial identity. So it's good that this information is here. There is discussion about his genealogy, and they present to us this extra pair paternity event, and they give us a date for when this happened. So an extra pair paternity event is basically the idea that the person that was thought to be I don't know if it's very correct to say the person, but the patrilineal, the paternity is not genetically what it was thought from the genealogy records. The other part that is very interesting is the risk for diseases that he had, and they bring to us these 2 genes here that account for a risk of liver disease. And there is an interaction between the genetic risk and alcohol consumption. I think this is a topic that is very interesting for everyone interested in genomics. And the other part that is interesting as well is that they do a isolation of hepatitis B virus, and they are able to well, first of all, that they are able to isolate that in a hair sample is something that is surprising and interesting, I would say, but they are able to propose a hypothesis that the interaction between the combination, let's say the combination between the infection and the genetic propensity that he has for developing a kidney disease is what caused him to, in fact, have a lot of gastrointestinal problems that is described for him. So that was the graphical abstract. Now a little bit of contextualization. Ludwig van Beethoven is a very influential and popular classical music composer. And this is information that the paper brings to us. He has health disorders that have impacted his career. Interestingly, I don't know if you all heard about this, but it's not the first time. I have always heard that he had hearing loss, which seems like it should be very important for his abilities to compose music. But we see that it did not prevent him from becoming one very important composer of classical music. He also had gastrointestinal disorders and liver disease. So why are we reading this paper? And so the answer to this, the answer that I want to give to this question is we are reading this paper because he requested that following his death, his disease be described and made public. The medical biographers proposed many hypotheses for his conditions, including heritable diseases. And so in the paper, what the authors did was they investigated the genetic and infectious causes of his disease. One thing that is interesting that I noticed in this paper is that they mentioned the improvement in ancient DNA methods, and that's my understanding is that that's improvement in extracting DNA, because these samples, I actually did not know that someone dying in the 19th century, right? I think he died around 1830. That's the 19th century. I think he died around 1830. I did not know that these DNA samples would be considered ancient DNA, because I thought only about things like 2,000 years ago, 3,000 years ago, I don't know, 10, 20,000 years ago would be considered ancient DNA. And again, that's because at least the first author is from the Department of Archaeology. So now we come to the figures. This figure conveys the idea of the provenance of samples, where the samples come from, who are the people that identified the sample, and there is a lock name here for the, I don't know if this is for the sample or someone that possessed the sample. But basically this is dealing with where the samples come from, whether it's authentic or not. They mention here a haplogroup. I don't know if I will exactly touch on the notion of a haplogroup by next week. But it's an easy way for you guys to understand a haplogroup is a group of mutations, or a group of variants or genetic variants that are inherited together. Okay. And then relatedness testing of the hair locks. I put a question in the notes here about relatedness, or a comment rather. Relatedness is a method to estimate relatedness of individuals because of their recent common ancestry. Two individuals are genetically related because they share a recent common ancestor, or that's basically what it is. They have a recent common ancestor. And so there are algorithms for calculating this. I'm not that much familiar with these algorithms, but this is to tell whether someone is genetically related to someone else. Apparently, these individuals here are identical twins. So the stump, I'm not sure how to pronounce this name, but my understanding is that this so stump is one of the hair locks of Beethoven. And I put the question here about Eert van Beethoven. This is a male ancestor that is mentioned in the paper. And I'm asking the question, could this person matter here? Because I'm basically asking, could the authors have figured out an answer to a supposed relative of Beethoven in his male lineage by doing this analysis? And I so far I was not able to respond to that question, but I'm just putting it there because I feel it's appropriate. And then the next figure, the figure 3 in the paper, is a picture of the stamp lock, is the stamp lock that was isolated from Beethoven. The authors conclude that this is a hair lock that is authentic, that it belonged to Beethoven, and they discuss how they came to that conclusion in that paper. I'm not going to go into that now. So yeah, so that basically with that discussion, we would have an answer for this question. How was this hair lock, this particular one? Because in the very first one, so that's this one, the stamp, that's the stamp. It's difficult to pronounce that. Why was this chosen? Basically, the question is, why was this chosen and not this or this or this? Well, it could not be one of these because they are not authentic, but why was this one? And how was it chosen, basically? The other question that I also ask myself is, how was this sequenced? Which part of the hair did they isolate? Because as far as I have read and known, it's my understanding is that it's the root of the hair that has some DNA that is sequenced. But then if this was cut, then it may not have the root. So I would ask myself these types of questions. And that's basically the question, how was the sample isolated for DNA sequencing? In other words, how do they know that this part has DNA from the person, from, in this case, from Beethoven, and so on and so forth. So these are just questions that I ask myself that I want you guys to think about to help you guys kind of understand the paper. It doesn't mean that I have all the answers now. Then in the figure 4 is the geogenetic triangulation of Beethoven's ancestors' locations. They do an algorithm to calculate that ancestry. It seems like they have used nuclear markers, and that's important for us because nuclear markers are not mitochondrial markers, okay? And by the way, the nuclear markers, it seems like you can use them to estimate you can use nuclear markers to estimate nuclear ancestry, and that's different from estimating the ancestry of a person, the ancestry of the patrilineal lineage of a person and the mitochondrial lineage of their ancestry. And then in the figure 5, it's a Y chromosome phylogeny of all the males tested. And so you can understand from this that because I mentioned all the males tested, and because I already mentioned the art von Beethoven, you can understand that the five samples here, VB1, VB2, VB3, VB4, VB5, are relatives of this person that it seems like from the genealogical ancestry, this person is in Beethoven's patrilineal ancestry. And that's how the authors were able to relate the, I'm gonna say haplotype, or I'm gonna say haplotype, or it could also be a branch, a phylogenetic branch. That's how the authors are able to relate the basically the genetic variants of this person with the genetic variants of the descendants. I think these 5 descendants are alive, and that's how they were able, well, by comparing, right? They were able to tell that the patrilineal ancestry of Beethoven does not seem to be the same as for these living male Beethovens, basically. I hope it makes sense, but if it doesn't, please read the paper, and we can discuss more about the methods in the following weeks. So basically, this was a phylogenetic analysis of the Y chromosome, all males, and it's very important to discuss the concept of an alignment. We're going to start discussing that next week. So I guess this question here, how does this inform about the paternity of a male, is just, I'm just basically asking you guys to think about how this figure can possibly inform the paternity of Beethoven's. Here I put some information about the living the genomes of the living individuals. I watched this video here about the racial identification of Beethoven. And yeah, okay. And then, so polygenic risk score is a concept that is important, but I'm actually not entirely sure if we will have a chance to discuss this with the algorithm that we're going to implement. But in any case, I think I'll talk about it a little bit. And that's this is the involvement of this gene that we can discuss a little bit more in the next slide, because in this slide they are calculating the prevalence of liver disease and cirrhosis. I don't know if it's cirrhosis or cirrhosis in English. Sorry for my bad English at this point. But what they're showing here is that there is an association between having heavy drinking and the risk for liver disease and cirrhosis. And thank you, Anna. Thank you so much. There is an association between high drinking and genetic variability in these genes. And finally, they plot a phylogeny of the hepatitis B virus that they are able to isolate in the sequencing. Because we are 5 minutes past the hour, I'm going to go straight to the conclusion. Well, the conclusion that the authors give, they say that the study illustrates the contribution and potential of genomic data as the primary source of historical biography, basically because they are from archaeology. Well, for me, I really like that this paper illustrates the heritable disease risk. I also really like that it shows the infection with hepatitis B. And I like the extra pair paternity event, not because that impacts the identity of this very important artist, but I like it to illustrate how we can computationally or scientifically detect such an event. Okay, so later I'm going to make sure I include those pieces of information in the conclusion. So that would be it. Now that you guys told me that it would be important for you if I gave you some more help in reading a scientific paper in general, not this one in particular, I will incorporate that for the future. But again, for Friday, we should do the survey. Actually, there's going to be a survey about the course and the presentations, and then we'll get started in the machine learning part to identify the neuroblastoma labels. Okay. Do you guys have any questions or any comments about the paper, by the way, or about anything? And if you, and I can always share the screen again. All right.
For the presentation, I'm
Mhm.
guessing we'll just be sharing our screens and then talking through the presentation.
Yeah.
Or do we have to submit the slides as well?
Oh, in the instructions, I told you to upload the slides in the drive. Please,
Oh, okay.
yeah, please put your slides in the designated folders.
Got it. Thank you.
Yep.
Are
Yes,
we
we start just answering Alexandra's question. We are going back to the normal time on Friday. Yes, it's going to be 4 for you guys in Santa Cruz. It's going to be 4 for you in Santa Cruz, right? Hannah, you're going to say something?
Yeah, I just kind of wanted to get like a timeline of like our due assignments. So it's like this week is presentations on commands or commands and um
Next week you can do a command again, or you can do a figure or a figure in the paper or a figure in the project. And by the way, if you guys want, what I would really like to do, I would be pleased to do, is I can talk to you in the mini project, because that's my research progress, right? So I really want to talk about it, because talking about it helps myself and also it can help you guys. Does it make sense? Yeah, but you can always you can present, you can choose. You can choose the command, you can choose a figure from a paper, or the project, or the mini project.
thank you, Professor.
All right. If you guys want, you can just call me Gio. You don't need to call me Professor. Gio is fine. I will do my best to, Victor asked when I'll post the recordings. I'll do my best to upload them by tomorrow. If I don't, please email me to remind me, or send a message on Slack. I know that I haven't uploaded the previous class, but I'll do my best to upload the previous one and this one by tomorrow. All right, guys, any other questions? Yeah, I will just also send a reminder that we are going back to the normal regular hours on Friday. And if I don't, please also remind me to do that. Okay. All right, no more questions. And it's more than 2 hours at this point. Thank you guys so much. Enjoy the rest of your night, and I'll see you again on Friday. Thank you.
Thank you. Bye.
Bye bye.
Thank you.
Bye.
Hasta
Thank you.
luego.